In this lecture, we're going to discuss DNA replication, which is the process by which DNA makes copies of itself. If you remember, during the first lecture of the course, we discussed the different phases of the cell life, and one of these phases function was to duplicate the DNA inside the nucleus in order to prepare the cell for mitosis, because we mentioned there are two different types of cell division. There's meiosis and mitosis. In mitosis, the amount of DNA in each cell is maintained constant, while in meiosis it is divided by 2, so you have half the amount of DNA. DNA replication is very important for cells that replicate, so it's important for mitosis. Semi-conservative mechanism has been proven to be the mechanism by which DNA replicates. In that mechanism, the parental strands are permanently separated, so we make a copy of each of the parental strand, and each of these strands binds to its complementary new copy, forming a duplex molecule with one of the daughter strands. As I mentioned, the older strands do not remain with one another, but they are distributed among the copies that are made from them generation after the next. In the conservative mechanism, the two strands from which we make copies remain together after the copies are made. So we have an old double helix that is the same as the one from which a copy was taken, while the new two strands bind with each other to make a double helix of their own. And in that case, that conservative mechanism is maintained. However, in the semi-conservative mechanism, we have one of each of the old strands bound to one of the new strands. It has been proven that the actual events that take place during DNA replication is the semi-conservative mechanism, so the old strands always bind to the complementary new strand. And this has been proven using nuclear tracking. As we mentioned when we studied protein synthesis techniques and protein characterization techniques, we said that any experiment that requires tracing how molecules are distributed and how they react over time, we needed a molecule that emits radiation. The same molecules were used to study the conservative versus the semi-conservative mechanism in DNA replication in order to trace where the parental DNA strands end up in, which showed that the semi-conservative mechanism was the one that is actually taking place inside our cells. The enzyme that does the job in DNA replication that connects DNTPs with one another is called DNA polymerase, because similarly to RNA polymerase, it polymerizes the DNTPs one with the other. Also, similarly to RNA synthesis, when we talked about transcription, DNA is replicated from the 5' to the 3' end in that direction. A phosphodiester bond is also formed similarly to RNA between the 3' oxygen of the growing strand and the alpha phosphate of the DNTPs. One of the major differences between DNA polymerase and RNA polymerase is that DNA polymerase requires a short pre-existing DNA or RNA strand in order to bind to them to continue their growth. It cannot start from scratch with no pre-existing DNA or RNA strands. These short DNA or RNA strands to which DNA polymerase bind to are called primers, and they are very important to begin chain growth in DNA replication. The location where DNA replication starts is called the DNA replication fork. Of course, as we know, DNA is a double helix, so it has a double helical format, and in order to be able to access one of the two strands, which is different from RNA, we need to unwind this double helix by an enzyme called helicases. We mentioned helicases before when we talked about protein synthesis. These are also used to untangle any secondary structures that are present in the mRNA. Helicases begin always at a special unique segment of the DNA parental strand. These locations are called the replication origin. These origins are very rich in AT sequence, so not GC, AT. And the reason for that is to unwind double-stranded DNA, as we mentioned before. We need to melt the DNA. In order to melt the DNA, we need energy. And we know that DNA with richer AT sequences require less energy to unwind, to separate the two strands from one another. So the replication origin is rich in AT sequence for that purpose. Once the helicases find that replication origin, they unwind the parental DNA strand. Then a special RNA polymerase, that polymerase's function is to deposit the short RNA or RNA strand that we called a primer. Hence, this RNA polymerase is called a primase. It forms the primer, which is a complementary sequence to the place where the replication fork is open at the replication origin in order to start the unwinding process. The primer afterward is elongated by a second enzyme, which is DNA polymerase. As we mentioned earlier, DNA polymerase needs to bind to a primer in order to be able to make a copy of the DNA. So after primase deposits a few RNA bases, polymerase comes in, DNA polymerase comes in and forms a new daughter strand after it binds to the primer. The region where all these enzymes and proteins get together is called the replication fork, as we mentioned in the previous slide. And replication proceeds, the growing fork and associated proteins move away from that origin as a new DNA is being copied. As you know, if you have two strings wound to one another and you try to unwind them, that is going to cause torsional stress. This torsional stress we have mentioned is relieved by the enzyme topoisomerase. If you remember, we mentioned there are two topoisomerases, topoisomerase 1 and topoisomerase 2. Topoisomerase 1 is present in non-bacterial cell, so in eukaryotes, and the purpose why we have topoisomerase 1 is that topoisomerase 1 cuts only one of the two strands in the double helix, so the chances of error are very, very low when the double-stranded DNA is ligated. There are two DNA strands, as we know from the double helix. One is called the leading strand and one is called the lagging strand. The leading strand is the one that can be used to synthesize the daughter strand in the 5' to 3' direction. So the leading strand, we have two strands, the leading strand and the lagging strand, and we can replicate both. We have to replicate both in order to make a whole copy of the DNA that is present. However, as you know, the two strands are connected with one another with different directionalities. So one is 5' to 3', while the complementary one is 3' to 5'. So only one of them can be copied in the 5' to the 3' direction. That one is called the leading strand. The other one, the complementary strand, is called the lagging strand. However, copying the DNA from the lagging strand is going to be a problem because we cannot grow the daughter strand in the 5' to 3' direction directly. So the answer to that is to synthesize a new primer every few hundred bases or so on the lagging strand, each of which is elongated in the normal 5' to 3' direction. So the solution is not to have the DNA strand grow from the 3' to the 5' direction, because that is impossible for DNA polymerase to do. However, the answer is to add a new primer every few 100 bases, and from that primer the DNA polymerase is going to grow the daughter strand in the 5' to 3' direction. Each of the segments that are grown are called an Okazaki fragment. The RNA primer for each fragment, because we said on the lagging strand we're going to have more than one fragment, is removed and replaced by a DNA chain from the growing neighboring Okazaki fragment. Afterwards, all these Okazaki fragments are bound with one another with a third enzyme, which is DNA ligase, that joins together these adjacent fragments. So if we look at one side of the replication fork, because we know there's a continuation on this other side, we have a leading strand and a lagging strand. Both of these are parental strands from which we are going to make DNA copies. The leading strand is one on which the daughter strand can grow in the 5' to 3' direction. As you see in the lagging strand, growing from this side as the polymerase moves is going to require DNA being replicated from the 3' to the 5' end, which is not an option. So the answer to that when DNA opens up, a new primer is added every few hundred bases, and a DNA polymerase binds to this primer and grows the daughter strand in the 5' to 3' direction. In the leading strand, we only require one primer to which DNA polymerase binds and grows the daughter strand in the 5' to 3' direction. In the lagging strand, we have more than one primer, and as we mentioned, once DNA polymerase binds to that primer, it grows the Okazaki fragments from the 5' to the 3' end. As it reaches the next fragment, DNA polymerase growing the previous fragment removes the RNA and connects the Okazaki fragment prior to it with DNA bases. However, the DNA bases are not going to bind together with the next Okazaki fragment. In order to do so, we need another third enzyme, which is DNA ligase, that binds all the Okazaki fragment one to the other. There are multiple replication proteins that are involved in the DNA replication process. The first one is the large T antigen, which is a hexamer. Hexamer is a word that means, if you divide it, hex means 6 in Greek, so it has 6 units. This large T antigen unwinds the paternal strand at the replication fork, so its function is a helicase that unwinds the double-stranded DNA. The primers for the leading and lagging daughter strands are synthesized by another protein, which is called, as we mentioned earlier, primase. This primase synthesizes short RNA primers. Then there are multiple DNA polymerases. The one that is involved in DNA replication is DNA polymerase alpha, short for pol alpha, which extends the RNA primer with DNTPs, forming a mixed DNA and RNA primer. So the primer is RNA that's connected to DNA, as we saw in the previous slide. The primer, as we mentioned, is extended into the daughter strand DNA by DNA polymerase delta, which has a proofreading mechanism that reduces the replication error. So pol delta checks the function of pol alpha and corrects any errors. Pol delta forms a complex with another protein called replication factor C, or RFC, and the proliferating cell nuclear antigen, a second protein, which is short for it is PCNA. These two displace the primase pol alpha complex after the primer synthesis, and they replace the RNA with DNA at the primer site. So this is a more detailed depiction of the replication fork. We have the large T antigen, which is the helicase. It moves in this direction. As it moves, it unwinds the parental double helical DNA. So we have two exposed DNA strands. The one that the daughter strand can grow in the 5' to 3 direction is called the leading strand. At the beginning of the leading strand, primase is going to have a primer of RNA bases to which pol alpha binds. Then pol delta binds to that strand using two different proteins, RFC and pCNA, and replicates the leading strand as it moves along the leading strand. At the lagging strand, we have more than one primer, as we mentioned earlier. So primase deposits the primer with pol alpha that extends the primer into a DNA, to which pol delta binds and extends the DNA further and replaces the primer that comes after it in the next Okazaki sequence. After this is done, a DNA ligase is going to connect the different Okazaki fragments at the lagging strand. The two proteins that we mentioned are pCNA. pCNA helps bind pol delta to the parental strand. pCNA has a hole in the middle of it. It has multiple subunits, and that hole allows a single stranded DNA molecule to pass through it in order to maintain the structure of the DNA. It maintains the double-stranded DNA inside of it as the new DNA forms in order to maintain its structure. We can notice here that there's another protein that we haven't mentioned, which is the RPA. RPA binds to exposed single-stranded DNA in order to prevent it from forming any secondary structure, so that the pCNA RFC pol delta complex can move along the single-stranded DNA without having any entanglements due to secondary structures. As I mentioned, pCNA is a homotrimeric protein, so it has 3 subunits that are all alike, hence the word homotrimeric. It has a central hole through which the daughter duplex DNA passes. This prevents the complex from dissociating from the template. After the parental DNA strand is separated into single-stranded templates at the replication fork, it's bound to multiple copies of the RPA, which is the replication protein A. This protein maintains the template in a uniform conformation. Then it is dislodged by pol alpha and delta. So its purpose is to prevent the formation of secondary structure of the single-stranded DNA that resulted from the T antigen helicase passing through that portion of the DNA earlier. So this is the pCNA. You see it has 3 subunits, hence it's trimeric, and these subunits are exact copies from one another, so it is homotrimeric, and it binds the newly formed double DNA strand. This is the replicating protein A, RPA, and it binds the single-stranded DNA in order to prevent it from making any secondary structures, in order to allow the pol alpha, pol delta, and pCNA to pass on the single-stranded DNA and replicate it easily. One of the efficient methods by which DNA replication is enhanced is by directionality, because we know that there is a replication sort of bubble, each of which has two replication forks, each on each side. they assemble at a single origin, then move in opposite direction, leading to growing the DNA strands in both directions. So as you see here, we have the replication origin, two helicases bind to that replication origin. Each of these helicases is going to move in an opposite direction than the other, and as it moves in that direction, it opens up the DNA double helix, allowing for DNA replication to take place. Eukaryotic chromosomal DNA molecules have multiple replication origins separated by tens of hundreds of kilobases. So you have replication taking place in different parts of the chromosome. There's a 6 subunit protein called the origin recognition complex, or ORC. This ORC bonds to each origin and associates with other proteins required to load the cellular hexameric helicase MCM, which is similar to the T antigen that we mentioned earlier, and whose function is to unwind the double helical DNA at the replication origin. There are 2 opposing MCM helicases that separate the parental strand at the origin of replication, with replication protein A binding to the resulting single-stranded DNA, as we mentioned earlier. The activation of these helicases is required to initiate cellular DNA replication. So since replication is a very important central pathway that takes place in the cell, it has to be highly regulated, and that regulation takes place by a specific protein kinase called the S-phase cyclin-dependent kinase. Once this kinase activates the MCM helicases, the S phase of the cell starts, and if you remember, during this S phase, the amount of DNA in the cell doubles, because the S phase is the phase during which DNA is replicated.